Hello, and welcome to Chemistry Review Video 3, where we will be discussing how things dissolve in water and the difference between acids and bases. The main take-home concepts from this video include understanding why a substance would dissolve in water, or why it would not, knowing the difference between a substance that is hydrophilic and one that is hydrophobic, and having a basic understanding of pH and why pH is important for microbial growth. So if we're going to talk about things dissolving in water, it is first very important to reiterate that water is made up of very polar covalent bonds. There's a strong difference between the electronegativity of the oxygen and the hydrogens, which means the electrons spend a lot more time around the oxygen than the hydrogens, creating a partial negative charge at the oxygen and partially positive charges on the hydrogens. This allows hydrogen bonds to form between the individual water molecules or between water molecules and other charged particles. We've already mentioned that salts dissolve well in water because the ions interact with the partially positive and negative regions of the water molecules, which stabilizes the charge, and that dissociates the positive ions from the negative ions in the crystalline structure. So when you add salt to water, it dissolves because the water molecules interact with the positively and negatively charged ions. But sugar is held together with covalent bonds. However, you can take a sugar cube and put it in water and it also dissolves. So what's happening here and how is that different than when salt dissolves? Sugar molecules stay completely intact when they dissolve into water. Each individual sugar monomer, however, has a variety of spaces over the surface of the molecule that have a positive or negative charge, and that's due to the polarity of the bonds between the oxygens and hydrogens around the molecule's surface. Because the molecule has regions on it that are charged, if you take sugar and you drop it into water, the positive and negative regions of the water molecules will interact with the positive and negative regions of the sugar molecules, and the sugars will then completely diffuse throughout the water solution. So while the sugar monomers themselves are not coming apart, they are separating out into the water solution and interacting with the water molecules. This is because sugar is hydrophilic. Hydrophilic means literally water-loving, and it occurs because polar covalent molecules and ionic compounds are made up of atoms with an unequal distribution of electrons. That unequal distribution of the electrons means that there are areas on the molecule or compound where you have a full or partial charge, and the polar water molecules can interact with them in solution, negative attracting positive, positive attracting negative. Hydrophilic substances will therefore dissolve in water. Other substances that are not hydrophilic would be substances that are nonpolar. For example, this is a molecule of mineral oil. Mineral oil is made up of hydrogens and carbons, all of which share their electrons very evenly. That means there are no areas around the surface of the molecule where charges build up, either positive or negative. So when you add any sort of oil to water, the two substances separate into distinct layers. That's because the oil does not have any area on it for the water molecules to be attracted to and interact with. So the water molecules attract to each other and not to the oil. It would be as if you took a bunch of small magnets and a bunch of wooden beads and mixed them all together. If you shook up the container, eventually the magnets would all stick together because they're attracted to each other. And even though they don't repel the wooden beads, because there is no attraction between the magnet and the beads, eventually the beads would all be in a separate layer from the magnets. And for this reason, oils are what we call hydrophobic. Hydrophobic literally means water-fearing. It's because nonpolar covalent molecules are made up of atoms that share their electrons evenly, so there are no charged regions on the molecule. Without at least a partial charge, there's no place for the water molecules to form hydrogen bonds to connect with the nonpolar molecule. So hydrophobic substances do not dissolve in water. Hydrophobic substances are things that are waxy or oily in nature, and those are always made up of primarily nonpolar covalent bonds. So when we talk about water molecules dissociating ions, one thing that water can do is dissolve water. The polarity of the individual water molecule bonds is so strong that out of every billion or so water molecules, you actually get some molecules dissociating into ions. So you have the hydroxide ion, which takes on a negative charge because it has an extra electron, and the hydrogen ion, which is just a proton that's lost its electron to the oxygen in the hydroxide ion. If you have pure water, there's always a very even ratio of hydrogen ions to hydroxide ions, and these are constantly reforming water molecules while other water molecules are dissociating. This is what we call a neutral solution. Now, when we start dissolving things into water, that may change whether or not the solution is neutral or not. For example, we can dissolve sodium chloride into water without changing its pH because the sodium ions and chlorine ions dissociate and interact with the water molecules, but don't change the ratio of hydrogen to hydroxide ions in solution. Something like hydrochloric acid, however, when it dissolves, it dissociates into a hydrogen ion and a chlorine ion. And that means there is an excess of hydrogen ions in solution, which makes this an acidic solution. An acidic solution is one which has more hydrogen ions in solution than hydroxide ions. The opposite can also take place. If sodium hydroxide dissociates into water, you get sodium ions and hydroxide ions, which increases the overall ratio of hydroxide ions in solution and makes the solution alkaline or basic in nature. A basic solution has an excess of hydroxide ions compared to hydrogen ions in solution. If we look at the pH scale, which ranges from 0 to 14, pure water with an even number of hydrogen and hydroxide ions is a pH of 7. As the pH decreases, getting closer to 0, you have an increased ratio of hydrogen ions in solution. So the more hydrogen ions there are compared to hydroxide ions, the more acidic the solution is, and the smaller the pH. Between 7 and 14, you have a basic solution. The more hydroxide ions there are compared to hydrogen ions, the more basic or alkaline the solution becomes, and the larger the pH is. Why is this important? Because as we mentioned in a previous video, there is a constant state of building up and breaking down components within our cells. These hydrolysis reactions and dehydration reactions utilize water and the hydrogen and hydroxide ions that water produce. So water is broken up into a hydrogen and hydroxide ion when a hydrolysis reaction occurs and a large molecule is split up into smaller pieces. Similarly, a hydrogen ion and hydroxide ion is produced when small pieces are linked together to create a larger molecule. This means that if you alter the pH of the solution and the ratio of ions in the surrounding environment, it'll change the efficiency of the reaction. So the biochemical reactions occurring within cells are very sensitive to the pH of the environment of that cell. And you can actually block cellular growth by altering the pH of an environment, something that we'll talk about in more detail when we look at ways to stop microbes from growing in places where we don't want them. Another aspect to keep in mind is that sometimes the byproduct of certain biochemical reactions is acidic or basic in nature. For example, fermentation, which is the primary way that microbes break down sugars for energy, releases acid as a byproduct of that chemical reaction. One way to test for microbial growth is to actually use a pH indicator that shows whether or not a particular sugar or food source is being used by the microorganism for food by looking for the acidic byproduct that is released as that organism grows. In this image, the organism growing at the top of the Petri plate is yellow because it's changing the pH indicator as it's producing acid as it grows on the sugar in the media. The pink organism that's at the bottom of the Petri dish is not producing acid, and even though it's similar to the other organism in many ways, it is easily distinguishable from the other organism by its inability to use this media as a food source, and therefore its inability to produce acid as it grows there. We'll discuss this more in depth both in the lecture and the laboratory as the semester goes on.